Today we're going to look at Module 5 over photosynthesis and cellular respiration. So let's start with reminding ourselves that energy is in the form of heat, light, and electricity, but energy is also stored in chemical bonds. And we can release that energy when we break chemical bonds. So the definition of energy is the ability to do work, and cells do work all the time. Cells actually perform three kinds of work: mechanical work, transport work, and chemical work. Mechanical work would be something like riding your bike and using your legs to push the pedals. But in cells, it would be something like beating cilia and flagella to move things past the cell or move the cell itself. It would also be contraction of muscle cells. And muscle cells use things like actin and myosin, really tiny fiber proteins, in order to cause contraction. Transport work, that would be something like active transport pumps, endocytosis, exocytosis, all of those vesicular transport examples we've looked at when we talked about types of active transport. And chemical work is biosynthesis, such as making polymers by putting monomers together. So here are the two chemical pathways we're going to look at: cellular respiration and photosynthesis. These are the two chemical equations for both of those processes. So for cellular respiration, glucose plus oxygen yields CO2 or carbon dioxide, water, and energy. Cellular respiration is essentially the splitting of glucose and the formation of carbon dioxide, energy, and water. We're really after the energy, so we would actually consider carbon dioxide and water in this chemical equation waste products. Carbon dioxide is something that we breathe out. Water is something we can find in our body. So cellular respiration is something that chemically occurs within us. That is why we breathe out carbon dioxide. That's why we're able to sustain a certain amount of water so chemical reactions can take place. And the energy created from cellular respiration is how we power a lot of cellular processes. And glucose being one of the reactants, that should be a key to you as to why carbohydrates and other nutrients are important for metabolism in human beings and other organisms. Let's look at the photosynthetic chemical equation now. It's essentially the reverse of cellular respiration. So we actually begin with carbon dioxide, water, and the energy here is sunlight. And those things, when they combine together or react as the reactants, can generate sugar and oxygen. So photosynthesis generates oxygen, whereas cellular respiration generated carbon dioxide. So they both generate different respiratory gases, and one uses energy and the other produces energy. So they are counterparts of each other, and they exist within a cycle. So keep in mind that we've talked about autotrophs. We've talked about autotrophs being the ones that can take sunlight and convert it into usable energy. They store that usable energy in glucose, and that's essentially kind of the mini version of photosynthesis. Plants can also perform cellular respiration, where they take that glucose and break it down to make ATP. So plants are interesting as autotrophs because they can perform both photosynthesis and cellular respiration. Whereas humans, we as mammals must basically take in our nutrients. We can't create our own. We can't convert sunlight into chemical energy. But we can use the nutrients we take in, monomers like carbohydrates made up of glucose. We can use that to make our own energy. So heterotrophs tend to only be able to do cellular respiration, whereas autotrophs can perform photosynthesis and cellular respiration. And we're going to see throughout this module that both the products of photosynthesis are the reactants of cellular respiration and vice versa, and that forms an energy cycle. So here's an example of that basic energy cycle. We can see at the top that plants are taking in sunlight energy. They're absorbing that energy using a specialized organelle called a chloroplast. So the chloroplast is the special organelle that contains a pigment to absorb sunlight. So that chloroplast is only found in plant cells. For the most part, any type of autotroph that is photosynthetic would have chloroplasts. Once that chloroplast in the plant here, as our example, has absorbed sunlight energy, it converts it into chemical energy. That chemical energy is in the form of glucose, and it produces oxygen as a byproduct. that oxygen can be used in aerobic cellular respiration. And aerobic cellular respiration primarily takes place in the mitochondria of both plant and animal cells. So here they're showing us the mitochondria in a rabbit cells. And mitochondria, as it carries out the process of cellular respiration, takes the glucose and oxygen, generates energy in the form of ATP, but also carbon dioxide and water as byproducts. And then those products from cellular respiration get reused in photosynthesis. So this is an example of an energy cycle between photosynthesis and cellular respiration. So we have to also talk again about cells and about how cells obtain energy. And you may think, oh, they make energy using mitochondria. Well, that's true. And that's why plant cells and animal cells have mitochondria. But what does the mitochondria use to make that energy? Well, it uses glucose. All cells can use glucose as a source of energy. They metabolize it, they break it down, and breaking chemical bonds releases energy in the form of ATP. Plants can actually convert glucose to sucrose or starch for storage, whereas we, humans, we convert glucose into long chains called glycogen. Sometimes we'll convert glycogen to fat, but we store glycogen in our liver and also our skeletal muscles. But these storage molecules are essentially going to be the bank or large amount of energy that can be produced if we break them down. So the storage molecules are or can be converted to glucose in a time of need to produce ATP for energy harvesting. So what is ATP? We know it's what the mitochondria produces. The mitochondria, as the powerhouse of the cell, is responsible for breaking down glucose and turning it into adenosine triphosphate, or ATP. So adenosine triphosphate is made up of an adenine base, 3 phosphates, and it's made up of a sugar, made up of a ribose. But it's relatively simple as a molecule. It plays a crucial role, however, in biological systems, including these metabolic pathways of photosynthesis and cellular respiration. So you can definitely think of ATP as not only energy currency of cells, but as essentially what cells use in order to carry out metabolism for day-to-day reactions. It supplies the power required for most cellular reactions. So here is that structure of ATP. So we can see that we have a 2-ringed nitrogen-containing base in pink called adenine. We have a ribose sugar. Then connected to the ribose sugar in blue, we have 3 phosphate groups. So phosphate groups are a phosphate atom attached to multiple oxygens, and one of those oxygens will be double bonded. So that's where we're getting the triphosphate from the 3 phosphate groups. So nitrogenous base adenine, sugar ribose, which is a pentosed 5-carbon sugar that we've seen in RNA, and then 3 phosphate groups, same ones that you find in phospholipids. ATP is also a part of the cycle. It's produced by cellular respiration and then utilized in photosynthesis. So its phosphorylation, or the addition and then removal of phosphates, is how it's cycled. If you break down ATP, you generally do so by breaking off its terminal phosphate, or the phosphate at the very end. And once that last phosphate is broken, we have a release of energy. But we also have a breakdown of ATP into ADP, or adenosine diphosphate, and one lone inorganic phosphate. So cells fortunately have been designed to convert ADP and an inorganic phosphate back into adenosine triphosphate, or ATP, by just adding an additional phosphate back onto the ADP, or adenosine diphosphate. So ATP must be recycled, and because it must be recycled, it becomes a part of this ATP cycle as it's kind of pushed back and forth between the metabolic pathways of cellular respiration and photosynthesis. So if we're looking at how cells obtain energy, they need ATP. That's the currency, but glucose is the savings. It's either stored as starch or cellulose in plants or glycogen in humans. So a lot of organisms have a savings of glucose or some type of metabolic bank. All cells use glucose as a source for their energy, but ATP is definitely more molecularly spendable. ATP can be phosphorylated or broken down to release energy in the form of broken bonds almost immediately if it's available, because ATP is designed to release just the right amount of energy needed for cellular work. But it doesn't empty the metabolic bank account because you should still have glucose in plants as cellulose or starch in storage, or in animals as glycogen. But the breakdown of glucose occurs in phases, so we'll have to look at this more later. And let's start by looking at the steps of cell respiration. Cell respiration is all about making ATP by breaking down glucose. And cells are going to use glucose and oxygen to make ATP. So we're going to break down glucose, but utilize oxygen in order to make spendable energy, or ATP. The byproducts of this process are carbon dioxide and water. The whole reason we call cell respiration oftentimes aerobic cell respiration is because it involves oxygen. A process that is aerobic involves or uses oxygen, or happens when oxygen is present. Something that would be anaerobic is essentially anti-aerobic, and so it doesn't use oxygen. So by saying that cell respiration is aerobic, we're saying it utilizes oxygen in order to break down glucose. So it definitely involves breaking down glucose, but only in the presence of oxygen. And keep in mind that cells are busy performing metabolic processes all the time. So these cellular processes like photosynthesis and cellular respiration, they're occurring constantly, as well as other processes that make proteins or carry out genetic expression. But many of these cellular processes require energy, so we need metabolic processes like cellular respiration to make ATP. Photosynthetic cells can make their own glucose, then break it down during cellular respiration to get their own ATP. But we have non-photosynthetic cells. We have to ingest our glucose or our nutrients that get utilized in order to make ATP. So glucose is the main carbohydrate of choice for making energy, or making ATP. So here are the three main steps: glycolysis, the citric acid cycle, also known as the Krebs cycle. This also includes acetyl-CoA formation, so we'll talk about that. And then the electron transport chain and oxidative phosphorylation. The electron transport chain with chemiosmosis is essentially called oxidative phosphorylation. But oftentimes books and textbooks, including yours, like to just focus on the electron transport chain and talk about the further processes required for this to complete. So let's look at where these processes occur. Glycolysis, the first step of cellular respiration, occurs in the cytoplasm of eukaryotic cells. Glycolysis is a two-part word, with glyc talking about the sugar or something sweet, and lysis talking about the breakdown or separation. So glycolysis really is the first step of cellular respiration, where we split and break down sugar in the form of glucose into two pyruvate molecules. When glycolysis is over and we have the two pyruvate molecules, if oxygen is available, we move to the other two steps and we move into the mitochondria to do so. If no oxygen is available, a process we'll talk more about later called fermentation takes place. So glycolysis has a meaning. It comes from the Greek glyko, meaning sweet, and lysis, meaning to split apart. So glycolysis, as the first step in the breakdown of glucose, does split glucose apart, and it splits it into two pyruvate. We've looked at where glycolysis occurs in eukaryotic cells. In eukaryotic cells, glycolysis occurs in the cytoplasm, but it also occurs in the cytoplasm for prokaryotic cells. Glycolysis is the only step of cellular respiration that does not require oxygen. So glycolysis does not require oxygen. Looking at glycolysis in more detail, we'll see that we're going to take a 6-carbon sugar, that's glucose, and separate it into two separate molecules. These two separate molecules, called pyruvate, will each have 3 carbons. In total, when we're done with glycolysis, we will have produced 2 ATP. We have to use some ATP in the process, So the 2 net ATP means the total left over and produced that we didn't have to begin with is 2 energy molecules of ATP. So this process consists of essentially 2 distinct phases. Again, we're going to break glucose down into pyruvate. 1 glucose molecule becomes 2 pyruvate. And the second phase is marked by the production of not only ATP, but the recycling and excitation, essentially, of NADH, which is nicotinamide adenine adenine dinucleotide. And this is an electron carrier. You can see that at the end of glycolysis, we're going to split these 6 carbons into 2 molecules that each have 3 carbons. So in glycolysis, we split glucose into 2 pyruvate, and throughout the process, we generate an electron carrier and energy. In total, we generate 2 net ATP because early on in the process, we had to utilize or use some ATP. So let's look at some overview. It occurs in the cytoplasm of the cell. Oxygen is not required, and it produces 2 pyruvate from an input of 1 glucose. It is anaerobic, meaning again, it does not require oxygen. And even though it utilizes 2 ATP, it puts 4 ATP out, which means we have a net of 2 ATP produced, and we're producing the electron carrier NADH. And we'll talk more later about what are the electron carriers for. All right, now how does cellular respiration extract energy from glucose? Well, cellular respiration also includes the citric acid cycle and the electron transport chain. And so we have to take those 2 pyruvate molecules and send them into the mitochondria. The chemical energy from the 2 pyruvate molecules is going to help us produce 32 to 34 more ATP as we go through step 2 and 3. The cellular respiration that occurs in the mitochondria, which we know is the powerhouses of the cell, they are going to produce from these 2 3-carbon molecules of pyruvate more energy by essentially breaking them down and producing a hydrogen gradient in parts of the mitochondria that we then pump through a specialized enzyme called ATPase or ATP synthase, and that's going to help us make ATP. So let's break down what's occurring. First, let's talk about what does the mitochondria look like. A mitochondria has 2 membranes. It has an inner and an outer membrane. Inside the inner membrane is a space called the mitochondrial matrix. Between the outer and inner membrane is another space called the intermembrane space. So here we can see an image of the mitochondria. So notice it has 2 layers, an outer and inner membrane. Inside the inner membrane, this space is the matrix. Between the 2 membranes, this space is the intermembrane space. And we can see what it actually looks like. So this is kind of an illustration. And down here at the bottom, we have a transmission electron micrograph of a true mitochondrion. So we can see outer and inner membrane, matrix, and intermembrane space. All locations you need to know for the mitochondria. So following glycolysis, which is step 1 of cellular respiration, we move into the citric acid cycle, also known as the Krebs cycle. This citric acid cycle does require oxygen indirectly, and it's going to occur in the mitochondrial matrix, so within that inner membrane. Before we can use the pyruvate molecules that enter a cell's mitochondria, we have to essentially modify it into something called acetyl-CoA. So the citric acid cycle includes this conversion of the 2 3-carbon pyruvate molecules into acetyl-CoA. Once acetyl-CoA has been produced, we will utilize it in the citric acid cycle, and it consists of 8 steps. So we're not going to go through each individual step, but during these mitochondrial reactions, we're going to see that we'll utilize carbon dioxide by producing it as a waste product, and carbon dioxide is going to come from the breakdown of pyruvate And that carbon dioxide will eventually diffuse out of this cell and into the blood, and that'll be carried to the lungs where it is exhaled. So if we look at the diagram below, we have a 3-carbon pyruvate. Remember, we would have 2 of these, so each pyruvate would be modified into acetyl-CoA. We would do that by having it lose one of its carbons and combining with some oxygen to form carbon dioxide. Then acetyl-CoA would enter the Krebs cycle and go through a series of processes that create some energy, or ATP, and also creates more electron carriers. It even gets rid of more carbon dioxide. So by the end of the Krebs cycle, we have converted pyruvate to acetyl-CoA. Then we sent it through the Krebs cycle, or citric acid cycle, and generated not only carbon dioxide, but energy in the form of ATP, as well as a lot of electron carriers. And we're going to use these electron carriers in the electron transport chain. Speaking of the electron transport chain, that is the last step of aerobic respiration. The electron transport chain also includes chemiosmosis, and those 2 steps together make up what we consider oxidative phosphorylation. This step requires oxygen. Oxidative phosphorylation, chemiosmosis, those terms are sometimes interchanged, but we're just going to think of the ETC, which is the electron transport chain, plus chemiosmosis, which is the movement of chemicals from high to low. Those are oxidative phosphorylation. So let's look at the electron transport chain first. This is located in the inner mitochondrial membrane of mitochondria. And the electrons that have been transferred from the electron carriers, NADH and FADH2, as they were picked up in step 1 and 2 of cellular respiration, are now going to be carried to the integral proteins of the electron transport chain. Because that's all the electron transport chain is. These integral proteins, or embedded proteins, in the inner mitochondrial membrane that transport collected electrons from one protein to another in order to build up a proton gradient in the intermembrane space. So the ETC facilitates the process of oxidative phosphorylation because it builds this chemiosmotic gradient in the intermembrane space. Then we can move that gradient of protons or hydrogen ions from the intermembrane space back into the matrix through an enzyme called ATP synthase. It's called ATP synthase because it synthesizes ATP, and it uses this passive transport of protons to do so. So oxidative phosphorylation is pyruvate breakdown and transfer of electrons along the electron transport chain, and the generation of ATP by chemiosmosis. So it utilizes not only the electron transport chain, but this process of moving protons. It occurs in the mitochondria. ATP synthase is embedded in the inner mitochondrial membrane, just like the integral proteins of the electron transport chain. The protons that are created are being pumped from the matrix of the mitochondria into the intermembrane space as the electron carriers move them across the electron transport chain. They will be used, the protons, to power ATP synthase. And ATP synthase actually takes phosphates and adds them to ADP, or adenosine diphosphate. And when we combine those together, remember, we get ATP, or adenosine triphosphate. So essentially, ATP synthase makes ATP. So ATP synthase, a synthesis of ATP, is what it stands for. It is the enzyme acting as a pump embedded in the inner mitochondrial membrane that makes ATP. During this process, oxygen will be the final electron acceptor, because what is occurring is we are transporting these protons along the chain into the intermembrane space of the mitochondria, and then oxygen will be there at the end of the electron transport chain to collect some leftover protons, and those 2 protons, which are essentially hydrogen ions, when they combine with water and some electrons, that will make water. So oxygen is the final electron acceptor, and in accepting that electron, it also binds to 2 protons, and those protons are in the form of hydrogen ions, and that will create water as a byproduct of cellular respiration. So high energy electrons must go across the electron transport chain. That's how all of this gets started. And the electron transport chain carries and releases these electrons as they move across, and a lot of that energy is given to hydrogen ions in the form of H+. Since a hydrogen ion only is made up of one proton and no electrons, we sometimes call it a proton. As these high energy electrons traverse the electron transport chain, we have to have that energy released in steps into the intermembrane space. And those small amounts of energy that we're losing at each stage of the electron transport chain will be harnessed again in that gradient of protons in the intermembrane space and eventually pumped through ATP synthase. And that buildup of protons in the intermembrane space is what is going to help us generate ATP. The high amount of protons in the intermembrane space moving into the matrix where there's less of them will be a passive process, and that passive process is called chemiosmosis. They can move back into the matrix of the mitochondria through ATP synthase, and that will help us generate ATP. Oxygen will be used near the end of the electron transport chain to accept some electrons and protons, and oxygen as the final electron acceptor will help us form water as a byproduct of cellular respiration. So one water molecule is produced for every 2 electrons that traverse the electron transport chain. Chemiosmosis, again, is the process by which energy is first used to generate a gradient of protons and then captures that energy of the gradient in the bonds of ATP as the protons flow down its gradient. So a way to simplify that is as we use the electron transport chain, energy is released. That energy is used to pump protons in the form of H+ into the intermembrane space of the mitochondria. Now there's a lot of H+, or protons, in that intermembrane space, and they can get back to the matrix or go from high to low concentration by going through ATP synthase. As they go through ATP synthase, the kinetic movement of the protons powers the ATP synthase pump to put an ADP and a phosphate or combine components to generate ATP. So the flow of protons, or H+, through the ATP synthase channel actually provides the energy to synthesize about 32 or 34 molecules of ATP for each molecule of glucose. And all that newly formed ATP leaves the mitochondria. It enters the cytoplasm, and it provides the energy needed by the cell. So without this continuous recycling, without the process of aerobic cellular energy, life would cease. People produce, use, and then regenerate the equivalent of their body weight of ATP daily. It's essential that something like cellular respiration can occur. That's also why it's essential for us to have oxygen in our body and eventually in our cells. In eukaryotes, the oxidative phosphorylation process that includes the electron transport chain and chemiosmosis takes place in the mitochondria, whereas in prokaryotes that don't have mitochondria, this takes place in the plasma membrane. So a couple of different locations there for where do these processes take place. So here's what the electron transport chain looks like. We have embedded integral proteins. We have our electron carriers bringing high energy electrons. It's collected throughout step 1 and 2 of cellular respiration. Some of the energy from these high energy electrons is lost as they travel through the electron chain. And that energy is used to pump protons as H+ into the intermembrane space. So they're leaving the matrix and going to the intermembrane space. In this intermembrane space, we build up a lot of these protons. A high concentration of hydrogen ions, or protons, is generated in this intermembrane space. As we get to the end of the electron transport chain, any leftover electrons combine with 2 protons still in the matrix and oxygen as the final electron acceptor to generate water, a byproduct of cellular respiration. Then, the high gradient of protons is utilized as a passive transport process to generate kinetic energy, and these protons move from high to low concentration from the intermembrane space of the mitochondria to the matrix to generate ATP. And ATP is generated by putting together ADP, or adenosine diphosphate, with a lone inorganic phosphate. And again, it's the kinetic energy of chemiosmosis, or the movement of hydrogen ions from high to low concentration, through ATP synthase that generates ATP. So what we can see here is the electron transport chain. It uses electron carriers to pump hydrogen ions or protons into the intermembrane space. We generate water as a byproduct because it's the final electron acceptor. And ATP synthase, which synthesizes or makes ATP, is powered by the chemiosmotic gradient or movement of protons. So electron transport chain occurs in the mitochondria, requires oxygen. And if we're looking at an overview, the electrons are transferred from electron carriers, NADH and FADH2, to the integral proteins of the electron transport chain and used to create a proton gradient in the intermembrane space. And the protons are then used to power ATP synthase, which makes ATP by adding a phosphate to it. So that's probably the most succinct overview you can get for the electron transport chain and the process of chemiosmosis. All right, so here oxygen was acting as a magnet for the flow of electrons through the electron transport system. So oxygen as the final electron acceptor is helping pull or direct the flow of those electrons through the integral proteins of the electron transport chain. And as the electrons are flowing, protons are being dispatched and trapped in that intermembrane space. So the proton gradient is important. It's important because we need that proton gradient to create the kinetic energy that turns ATP synthase like a turbine, and that turbine or motor movement of ATP synthase is actually what is used to make ATP, or combined adenosine diphosphate with inorganic phosphate. So here's another overview slide, just like we had for glycolysis. The inputs are electron transport carriers carrying excited electrons, so NADH and FADH2. The outputs are water and 32 to 34 ATP per glucose molecule total. The electron transport chain is located in the inner membrane of the mitochondria. That's also where you'd find ATP synthase. The chemiosmotic gradient of protons builds up in the intermembrane space. And we're going to pump the protons from the intermembrane space back into the matrix in order for chemiosmosis to take place and help us generate ATP with ATP synthase. So let's look at that in kind of a general overview. Our steps that we've talked about are glycolysis, the Krebs cycle, also known as the citric acid cycle, and the electron transport chain and chemiosmosis, collectively known as oxidative phosphorylation. We can see from glycolysis we got 2 ATP. From the Krebs cycle we also got 2 ATP, but we get the most from oxidative phosphorylation, which includes the ETC or electron transport chain and chemiosmosis. So we can get anywhere from 32 to 34 ATP from the electron transport chain and chemiosmosis. So the general total given in most books is 36 to 38. Here with this example, we have 36 ATP total produced from aerobic cellular respiration. Cellular respiration can also extract energy from a variety of foods. We don't have to use carbohydrates. We can use proteins, we can use fat, and we can use those things also from the diet. So there are intermediate molecules that can enter certain parts of the cellular respiration pathway. We don't have to begin with glucose. For example, amino acids of proteins can also serve as an energy source. Fats are also an excellent source of energy. They may serve as a major energy storage molecule in animals, but they can be broken down into acetyl-CoA, and acetyl-CoA can enter the Krebs cycle. So a limited intake of fats will actually allow this process to happen. Will allow cellular respiration to utilize fat for this process, or essentially break down fat. But the overindulgence of fats is actually what causes the body to store excess fat, and that's when it's not used for cellular respiration. So here you can see how we might break down food into its monomers and then use it for either glycolysis or at the pyruvate stage, or have already converted something to acetyl-CoA, which enters the Krebs cycle. And that will complete then with the electron transport chain and chemiosmosis, just like the example we were looking at, starting with glucose. So glycogen from the liver and muscles together with fats, they can feed into the catabolic pathways for carbohydrates. They just might not start with glycolysis like we did when we started with glucose. So some of these other nutrients can also help us create ATP. And glycolysis is used by virtually every organism on Earth. Under aerobic conditions, when oxygen is available, cellular respiration normally follows glycolysis. But there are earlier forms of life that didn't have oxygen available. They performed anaerobic fermentation after glycolysis. And some microbes even still lack the enzymes for cellular respiration, so they rely solely on fermentation. There's actually various microorganisms still thriving in places where oxygen is limited or absent. So aerobic respiration uses oxygen as the final electron acceptor. When there's no oxygen, we don't have it as the final electron acceptor, so we have to perform a process that essentially recycles the NADH, or the molecule we are considering an electron carrier. So the collective term fermentation describes a process that uses an organic molecule as the final electron acceptor instead of oxygen. Fermentation is just a cellular process used to produce ATP without oxygen. It doesn't generate as much ATP as aerobic cellular respiration, but it can generate some when oxygen is not available. Many prokaryotic organisms use anaerobic respiration, and some can even switch from aerobic to anaerobic based on their environmental conditions. So let's look at fermentation. So there's actually two types: lactic acid fermentation and alcohol fermentation. Some tissues in animals and some strains of bacteria actually use lactic acid fermentation. For example, in the muscles of animals, a form of respiration that is anaerobic occurs when muscles are fatigued, or when someone's exercising, for example, so hard that their muscles are contracted and blood cannot get to that area right away. So fatigued muscles, or ones that are lacking proper blood flow for a moment, they may use this lactic acid fermentation to make sure if they can still get ATP. So this is one way to effectively provide energy to the tissue without oxygen present. The other form, alcohol fermentation, yields ethanol, which is one product of alcohol fermentation, but also yields energy. So fermentation produces either lactate or alcohol, and sometimes carbon dioxide. It also produces 2-net ATP because the process of glycolysis still occurs. But there is going to be just 2-net ATP produced. No energy is produced from the actual process of fermentation that is going to be used to regenerate NAD plus from NADH. So fermentation does not produce more ATP that the body can use as a reserve, but it is necessary to regenerate NAD plus. And in order for glycolysis to occur and create that just minimal amount of 2-net ATP, that's what fermentation occurs for. So glycolysis is still the step that generates ATP. Fermentation is just the recycling process to make sure glycolysis can continue. So let's take a closer look at lactic acid fermentation and alcohol fermentation. So some cells ferment pyruvate to form lactate. So lactate fermentation produces lactic acid from pyruvate. So we take the pyruvate from glycolysis and kind of turn it into lactate. Lactic acid can actually make muscles sore if it builds up. But we also use lactic acid in other types of fermentation processes, such as converting milk into yogurt or sour cream and cheese. So a lot of bacteria is used in order to do those types of conversions, because bacteria are going to be the type that are anaerobic and can perform lactate fermentation or produce the lactic acid needed for those types of food conversions. But lactate fermentation itself is just there to regenerate NAD plus from NADH, which is produced at the end of glycolysis. In the absence of oxygen, pyruvate does have a different fate. If we can't break pyruvate down further into acetyl-CoA and have it enter the mitochondria, that's when you would need fermentation to recycle NADH into NAD+. And if you've ever experienced the burning sensation in your muscles because you've overexercised, maybe the day of or the next day, that's because you had too much lactic acid buildup that couldn't be broken down in your overexerted muscles. And because you didn't have oxygen for a moment, because maybe you were overexercising, that lactic acid was produced as opposed to energy from aerobic cellular respiration. That means fermentation occurred in your own body. Even some organisms, like yeast, can survive using anaerobic respiration for a very long period of time, and that is the basis of beer and winemaking. So that brings us to some cells engaging in alcoholic fermentation. For alcoholic fermentation, the two products are ethanol and carbon dioxide. And this type of fermentation is still just to regenerate or recycle NADH back into NAD+, so glycolysis can keep going, so we can release or create at least some ATP, even when the entire process of aerobic cellular respiration cannot occur. So here is what lactic acid fermentation looks like. We take a 6-carbon glucose and break it down into 2-pyruvate. Because there's no oxygen present, we have to do fermentation, which doesn't produce energy, but it does recycle NADH back into NAD+, and we'll use NAD+ for glycolysis to continue again, and that helps us keep regenerating 2 net ATP from the process of glycolysis. In alcoholic fermentation, the products are 2-ethanol and 2-carbon dioxide, and fermentation again here does not produce any energy itself, but instead recycles NADH back to NAD+, which is used to make sure glycolysis can continue on, producing 2 net ATP in the absence of oxygen. So fermentation does play a role in the human body and other organisms that can also perform cellular respiration, but mainly in the human diet, we've used it to make certain foods like yogurt or wine or beer or cheese. It's also used in baking. The CO2 that is produced in alcohol fermentation is actually used to help bread rise. So here are just some of the foods that use fermentation in order to be produced. So you can see here the cheese and the bread both use different types of fermentation, one lactic, one alcohol fermentation, in order to be produced to the method or variety that we are accustomed to. So next class, we're going to look at photosynthesis. So we're going to look at how plant cells can break down the glucose they made their own into ATP. We're going to look at how do they make that glucose themselves. So how are they converting light energy into chemical energy?